Thank you, madam. Okay, so good morning, everyone, again. So in the last class, we have had a look at the xylem tissue, which was one of the complex permanent tissue. Yeah, so I'll not go back to again that description, what is complex permanent and so on. But just because of their different structures present within in it, they are part of complex permanent tissue. So that is xylem and phloem. So we had started with one of it, that is xylem, which was majorly responsible for conduction of water and minerals or the salts of it. Then there were different terminologies that we had looked at, that is primary xylem, secondary xylem, and like we had seen, the primary xylem actually originates from the procambium, which is the earlier version of the cambium, and secondary xylem majorly originate from the vascular cambium, that is already grown part of the tree, which is like old enough, so it will have vascular cambium, and from it the secondary xylem will grow. Now, in both of these cases, primary as well as secondary, we will have two different types. One which is protoxylem and the other one is metaxylem. Protoxylem is nothing but the earlier version of xylem, right? And like it's like you might wonder then what is the difference between primary xylem and protoxylem. But within primary xylem, when the cells grow at the start, they will be protoxylem, and then it will go into the metaxylem of the primary xylem. And once the plant has sufficiently grown, it forms those vascular bundles which has cambium. So from that, whatever xylem arises, that will be first forming protoxylem and then metaxylem. So that's how the arrangement is. Then we had looked at this tracheid structure, which was the major conducting element. It has these different septums at end of each cell, where two tracheid cells they are connecting, and there will be a transverse septum, and which is not really continuous. It is kind of porous, so that water can move through uh, these pits or through the septum, which is porous in nature. Then the vessels, again, this was like another conducting element or conducting like part of the xylem, and it is like kind of similar to your tracheids. Just the difference was in terms of the thickness and the diameter of these two different tissues. So this one is the xylem tracheids, this one was the xylem vessels. And the fibers, like they were just the supporting structure around these tracheids and vessels, and the xylem parenchyma, which was more responsible for the storage of certain materials, including starch, fats, and tannins, and so on, and also responsible for radial conduction of water. There always had been a tricky question being asked around this, which is the xylem component responsible for radial conduction. Now, what we usually remember is conduction of water, xylem. So, like then we just tick on something which is xylem, or we write only xylem. But when we talk of radial conduction, like I said, across the radius of the xylem tissue, then it is xylem parenchyma specifically. So again, this is how actually it looks like in the like reality. So these bigger ones, these are nothing but your xylem vessels. The smaller ones, these are nothing but your tracheids. And these kind of tissue which you see here, they are xylem fibers. Like you can see it here, these are the xylem fibers. And like xylem fibers, they are associated with the xylem parenchyma. So that's the overall arrangement of the xylem tissue in reality. Okay, moving on to the next topic, that is phloem. Now again, if you look at the overall xylem structure and phloem structure, they will not be much different, because both of them, they are conducting tissue, right? One conducts water and salt or minerals, that is xylem, and the other one conducts water along with the food material. But water, like not necessarily all the textbooks will mention it. Reference book they do mention it, because the food of plant is glucose. Now you don't have a bags or something, some material which will carry glucose from one part of the plant to the other part. So glucose is always conducted in the dissolved form, that is with water. So even if it does not mention that phloem conducts water and food materials, it ultimately means the same thing. And the reason I said is because the food material, they are dissolved in water. So obviously water also has to get transported to transport the food materials. And usually this happens from leaf to the other plant, other parts of the plant. Now, why is that so? Can someone tell me why the phloem would conduct the food materials from leaf to the other parts and not any other way? Someone, before I take someone's name.
So could you repeat the question once?
I said, why would the phloem conduct the food material from leaf to other parts of the plant, and why not other ways?
Sir, because leaves produce food.
Raise your hand.
Yeah, because leaves are the one who capture the.
Guys, raise your
Guys,
hand,
yeah,
sir.
raise your hand.
Yeah, I was just about to say that. Okay, maybe someone who has raised hand first. Shrihita did it. Okay, yes, Shrihita, say it now.
So because the photosynthesis happens in the leaves, no, sir.
Yeah, so that's correct. But then imagine the case of the transporting the food from leaf to the roots, right? Obviously, yeah, as you all know, the for food or the photosynthesis process happens in the leaf structure, and from there all the food that is being carried in the in the phloem of the leaf, then it comes to the phloem of the stem, and then from this phloem of the stem it goes to the roots part, and so on. Like not only it goes to the roots part, but on this process of transport or on the way, the phloem will also distribute to those parts through which it is getting transported. So that is the obvious reason why it happens that way. Major of this food material usually goes to the growing regions, because obviously they are the ones in the need, and they don't have their machinery uh, as of now, and that is why the growing regions will always need more and more food material. Okay, now on the basis of origin, phloem is classified into two categories, same like xylem. How it was primary xylem and secondary xylem, phloem is also the same, that is primary phloem and secondary phloem. Nothing different there as well. Now, primary phloem will originate from the procambium, again the same thing. Are you, I hope you are getting the similarity. Whatever we had seen earlier in terms of xylem is exactly same here as well. Just there is a difference of names in term name and obviously the function in reality, that is xylem and phloem. No, you are just looking at me, so I was worried. Fine. So sorry, there is someone in my class, and that's why I'm laughing in between. There is similarity with respect to the xylem and phloem. So there is primary phloem which will originate from procambium, and the secondary phloem will originate from the vascular cambium. The reasons remains the same. Pro refers to earlier version of the cambium, that is when the plant had just started sprouting or started growing. So that's when whatever cambium has formed, that will give rise to the primary phloem part here. And as and when it grows further, that is vascular cambium will be formed, and that vascular cambium will have secondary phloem. Now, on the basis of development, Again, primary phloem is categorized into Proto Phloem and Metaphlloem. This is again the something similar to your xylem. And the Proto Phloem has the narrow sieve tubes. Again, this description we will see later when we see the components of phloem. Xylem had components of tracheids, vessels, fibers, and parenchyma. 4 components. Similarly, phloem will also have certain components in terms of tubular structure, fibers, and parenchyma, which we are going to see further. And those tubular structure, in case of phloem, they are called as sieve tubes. Whereas the metaphlloem has a bigger sieve tube, and protophloem will have a little smaller diameter of those sieve tubes. And reason is again very logical. Protophlloem is the earlier version of the phloem, right? So that means it is not yet completely grown. So obviously its diameter has to be little smaller. While on the other hand, metaphlloem has sufficiently grown, or it is in the older parts of the tree or a plant, and that's when it will have bigger sieve tubes. So it's again a very logical description. Phloem will remain active for less duration as compared to xylem. Now, can someone answer to this question? Why phloem remains active for less duration as compared to xylem? Here.
Because photosynthesis only occurs in the day.
Only occurs in a day.
No, like when the sun is there in the day.
Okay, what if I question that statement? I say photosynthesis occurs for 24 hours. Means which we are going to study. There is something called as light photosynthetic reaction, and there is something called as dark photosynthetic reaction, which completes the cycle of photosynthesis. So in a way, the statement that you said may not be completely true. But you are right, for photosynthesis, sunlight is required. So that statement is perfectly right. But otherwise, no. But any which way, like maybe you can complete your statement. Why would phloem remain active? That the answer should still remain the same, I feel. Here.
I mean, in 8th we were told that, seventh we were told that cellular respiration takes place at night. So maybe that's why.
Yeah. So like I said, there are light and dark photosynthetic reaction, which we'll see in one of the chapter later in plant physiology itself. So like first half of the reaction of the photosynthesis happens in the sunlight. When the sunlight is there, that is the daylight or day time. And the later half of the reaction, when the photosynthesis reaction has to be completed, that happens at night because it doesn't need sunlight as such. But yeah, like you said it correctly. So the food is getting synthesized during the daytime, right? But not necessarily photosynthesis is happening in the daytime. Photosynthesis will complete its reaction by the night also. And so it's a continuous process as such. But then majorly, the first part of reaction, which is generating the glucose, happens during the daytime, right? While if you compare, that means the food has to be transported to all the other parts of the plant only as and when it is prepared, right? It's something like your mom will serve the food as and when it is done. She doesn't give you the food every now and then. She will say chuk bato abhi, right? But on the other hand, with respect to xylem is conducting the water and the minerals which are required throughout. Now there is a process in plant that happens, that is transpiration, and this is equivalent of sweating in human, right? Now the moment it is equivalent of say sweating in human, that means plant also do transpiration, which is loss of water. That means they need more and more water every now and then. They may not need more food because there is no loss of food, but there is surely a loss of water. So xylem has to be always active every now and then to transport the water and minerals and salt from the soil. But phloem will only remain active for less duration. As and when the food is prepared by the leaf structure, it will get transported slowly to different areas of the plant. So I hope this part is clear. Does anyone have any question here in this aspect of the phloem? Namit, would you have any question?
No, sir.
Soham?
Nah.
Okay, so fine. So I assume that most of you have clarity of the basics of phloem, right? Now, looking at certain characteristics of phloem tissue in general. Now, phloem consists of 4 different types of cell. Actually, let me discard that statement because we will see those 4 different types of cells or tissue in the later part of the slide. So, like, just forget that. So, in general, the sieve cells or sieve tube. Now, again, I assume everyone knows what a sieve is, right? It's a kind of filtration system that we use, like whenever we are at home, like mom use it for sieving atta or flour or something like that, or even for grains it is being used. So sieve is kind of a filter which separates few things. Now same analogy works with the phloem also. These sieve cells or sieve tubes, these are the tube elements. Tube elements means these are a tube like structure which are living because their cells are living and they are thin walled. Why they would be thin walled? Again, you can logically try to recognize this fact. They should be thin walled because there has to be certain transfer of material across the membrane of these sieve tubes or sieve cells. Now, why they are differently called? Because they are present in different types of plant. The major category of the plants is gymnosperm, like and angiosperm, the flowering and non-flowering types of plants and so on. Right? We need not go into those details. I'm sure this you have learned in last year in the plant kingdom classification. What is gymnosperm and what is angiosperm? If not, you can just quickly Google it later after the class and you will understand the difference. It's very simple. So these sieve cells which are present in gymnosperm and sieve tubes which are present in angiosperm, they have these tube elements or tubular structure which are living in thin wall. Now a matured sieve tube, now I will not say sieve cell every time because it's the same and we are majorly be talking about the flowering plants. So a matured sieve tube elements lack nucleus and thus they are enucleated or no non-nucleated living cells. Now you might wonder how they are like living and yet having no nucleus. Now that is something which is living does not necessarily mean that it has to have a nucleus. Living also means that it is doing certain metabolic functions with respect to certain proteins and enzymes. That can also make it a living. And also there is always an exchange of some of the genetic material that keeps on happening across the cell. So whatever is required for one cell which doesn't have nucleus, can get it from the other cells, neighboring cells, for example. Yes, Ruchira, you have a question.
Yes, I said, does the lysosome eliminate the nucleus?
Not necessarily. Like for example, your RBCs in your human body, also they don't have any nucleus as such, right? But they are also called as cells. It's something similar. That means they are functional unit responsible for performing certain function. They do not have nucleus because while they are being so these are the cells which I said these are differentiated over the time, right? So the nucleus, whenever it is not required, and whenever the cell has sufficiently grown, that will be developed in that process itself. So nucleus need not, so there are something called as unequal divisions. Like you would probably have heard of terms mitosis and meiosis, which is cellular division process, and which are almost equal division processes, right? But in some cases there are unequal division processes which does happen, and these unequal processes of cell division, they give rise to these kind of cells. So one cell will have nucleus and the other cell need not have the nuclear part at all, and it will just have some part of the cytoplasmic elements. So that's how they are formed. Not necessarily your lysosome has to degrade it, and so it has disappeared or not present and so on. Yeah.
Yes, sir.
So that is the these cells of the sieve tubes. They are non-nucleated cells. The central vacuole is present in each of the sieve cells. Now, obviously, the sieve tube elements. Now, because these are like kind of conducting tissue, and they need to have enough space within the cell, vacuole acts as the storage unit, and they have certain vacuoles present in these sieve tubes. The cytoplasm of the sieve tube element show cyclosis in the form of thin layer around the vacuole. Now, you might see what a complicated statement, but not so complicated. It just around the vacuole, there is a thin layer. And how it is formed? It is just formed by the cyclic reaction of certain biomolecules, and then it will form a thin layer around a vacuole. So that's how it is. Again, vacuole is the three-dimensional structure present in the cell. So it's like a balloon which is hollow inside, but vacuoles are not necessarily always hollow. They might have stored certain water, certain elements, sometimes food, and so on. They have different functions depending on what type of cell it is. So vacuole will have an additional thin layer which is formed because of the cyclosis, right? Cyclosis is nothing but the cyclic reaction process of the various molecule, and that's how the thin layer will be formed around the vacuole. In angiosperm plants, the sieve tube element, they are arranged with their ends from the ends and form the sieve tube. Now this is something same like tracheids also. Like you have let's say 1 feet of tube each, and then you keep arranging one on top of other and also like next to it, right? And then what you get is ultimately the arrangement with the end-to-end arrangement of the sieve tube. Let me see if I have diagram here. Yeah, let's assume this is one sieve tube and this is another sieve tube. So at their ends are kind of matching with each other, and that is what it means by the end-to-end arrangement. Yeah, so, and that is the case with the angiosperm, but even in the gymnosperm, the cells will be arranged in the similar way. Yeah, it wouldn't be that different, because ultimately they have to form a cavity or a lumen of the tube to conduct the water portion. Sieve plate, these are the oblique transverse perforated septa. Now again, same like how it was present in the tracheids as well, right? There was the transverse septum between the two individual cells, and these were also those were also perforated, and these are also perforated. That means they are porous in nature, and they are present in between the two sieve tube element at their end wall. Now again, here what you see here, this part is nothing but the sieve, which is kind of shown here, which is perforated. Yeah, that means it has certain holes or pores. It is porous and materials are transported through these pores. That is very, very obvious statement. I will just finish this part and then I'll take if you have any question. Now, this is not the part that is actually in your syllabus, but I thought it will be important to know certain extra facts about it. So that's why I have kept it here. So do not get overwhelmed if you don't understand this part. But yeah, do ask questions here if at all. So callous is nothing but one of the carbohydrate type material. Like you have glucose, fructose, lactose, galactose, all of these. Callous is something like that, is deposited on the radius of the pores. So we have this radius of the pores, and the callous will be deposited around these pores. So that's what it means. So callous deposited on the radius of the pores during the dropping season, that is during the autumn of the leaves, to form a thick layer, and this is what is called as callous fat. Now what will happen is, so this callous material will be deposited on this pore, and it will kind of like uh, block, but not completely block. So slightly it will block these pores, and that will form a callous pad. So because it is forming a layer of callus on whole of this sieve plate, it will form this callus pad. Now what is the function of this callus pad? So the sieve plate is actually protected by the callus pad. How? By preventing from bacterial infections and drought. Now, whenever there is a drought, less water, in each of the sieve tube, the water will be stored. It's nothing but, let's say you have attached a pipe to the tap, and then you close the tap and you take out the pipe. The pipe has certain amount of water, right? If you are not draining it out completely, pipe will always have certain amount of water, and that is how the sieve plate will help in protection, help the plant from the drought. And with bacterial infection, because it will not allow the transport of bacteria across the plant, so these cells or these sieve plate structure, because of the callus pad present on it, it will prevent the bacterial infections as well. So that is the general function of the callus pad. And again, whenever the spring season is back after the autumn, the callus will dissolve and the regular movement of the water will start. So that is the general reason why the callus pad is present in those particular plants. Now in gymnosperms and pteridophytes, again pteridophytes is one of the category of the plant. So like you need not worry about the names, but in these type of plants, the sieve cells do not form the sieve tube, and they are arranged irregularly. Like what we saw here, the sieve tube cells, they are arranged in the tubular format, one on top of each other. But in these two types of plants, these sieve cells, they are not necessarily arranged to form a sieve tube like structure always. They are arranged little irregularly, and the sieve cells, they have sieve plates on their lateral walls. So instead, let's assume what you have done is you have like lot of big spheres, like let's say balls, you have kept filled in certain structure, and those balls, they have perforations or they have holes on their walls. So that is what is the sieve cell will have sieve plates on their lateral walls. So the spherical wall having perforations on its walls, and through that the conduction of the water along with the food material happens in a zigzag manner. I hope you can assume this scenario, how it would happen in the gymnosperm and pteridophyte kind of plants. But ultimately that forms a kind of capillary network, and then those capillary network will they will be responsible for transportation of the food. So that's not really like there are no tubular structure forms. The capillary type of tubular network, they will always be formed. So yeah, in angiosperm, on contrary, in that is the flowering plants, the regular dicot flowering plants that you have, the food conduction is erect and efficient. Erect in the sense it happens in a straight way, either from top to bottom, that is from leaf to the root part. So that way it is erect and it is very efficient because the path which is crossed by the food and water is short, and that is why it is more efficient. Sieve element, they contain special type of protein, P phloem. Now again, this protein is responsible for protection of these sieve tubes or sieve cells, and that is what is the function of this P protein. So that is the additional information we have with respect to the phloem. Now, okay, do you have any question here? And I hope it wasn't too much to take in one go. Okay, so if there are no questions, so this is how a general phloem would look like. Again, these are the phloem tissue, and this is the cross section, that is the longitudinal section and cross section. You can just go over this description, you will understand what they are talking about. I need not really explain each and everything here. This is just how it will look in reality. These will be certain tubes which are perforated at their end wall, and they will be conducting again something similar to your xylem itself. I guess if I don't know if we have ready-made slides of these, but if at all you guys come back to the campus, we'll try to look at the xylem and phloem tissues of certain plants. It will be very interesting to see them in reality rather than just in these photos. Okay, so then coming to the next component of the phloem, that is companion cell. Now these are again companion, the name itself would say something about them. So these are companion for something in the sense in the phloem tissue, they are acting as a companion for the some structure, and that's why they are called as companion cell. So these are thin walled living cells, and the sieve tube elements, that is the sieve tube structure that we had seen earlier, and the companion cells, they are connected by the pit fields, that is the holes present in their longitudinal wall. So if you look at these are the companion cell, and these are the sieve tubes. So this bigger one is the sieve tube, and next to it is your companion cell, which has a kind of pits, right? And on their longitudinal wall, that means on their vertical wall, and which is common wall for both, that is for the sieve tube as well as for the companion wall. So this wall is common for the sieve tube structure, which is long tubular structure here, and the short tubular structure, that is the companion cell. And usually it is being said that if one of these either sieve tube or the companion cell is dead, the both of the or the other counterpart will also die. So that is the actual true case, because the nutrition and everything that will not be transported to the either of the cells, and then obviously they will die. The companion cell is laterally associated with each sieve tube element. So each of the sieve tube that you see will always have one companion cell along with it. And these, whenever they are forming together, when the phloem was about to form, these sieve tube element or sieve tube as well as the companion cell, they will originate together from a single cell, and that's why they are also called as sister cells. Like we already know the process of mitosis, I assume so, and one cell giving rise to two cells. So one cell will form sieve tube, and the other cell will form the companion cell, and they will stick together and stay together forever. So that's what is these companion cells also being called as sister cells. Now the companion cells as well as the sieve tube structures, they will maintain the close cytoplasmic connection with each other through plasmodesmata. Now because they have common wall, like you can see here, they have a common wall, and also at some time point there will be this or some structural point there will be plasmodesmata structure. These are again nothing but like internal pipeline. Like let's say we have neighboring houses and we just open up a small portion of the wall just to exchange something either from that house to this house or from this house to that house. That's exactly how plasmodesmata work between two cells. Two cells when they are very close to each other, and in this case they have common wall. So in that common wall there will be certain tubular structure which will allow the transport of certain material through the cell wall of the cell, and that is how this plasmodesmata would work there. Companion cell is a living cell with a large nucleus, and on the contrary, the uh, like we had seen the sieve tube structures, like here we had seen that. Where is it? Where is it? Where is it? Yeah, the sieve tube elements lack nucleus. So you remember I spoke about the unequal division arising from the single cell. So one cell will take the nucleus and other cell just takes some part of cytoplasm and forms the structure, and that is what is meaning of this statement, that is companion cell is a living cell with large nucleus, and because it is a sister cell of the sieve tube, it will not it doesn't have the nucleus, and that is a type of unequal cell division, unlike mitosis or meiosis. This nucleus also controls the cytoplasm of the sieve tube element, and I also mentioned this, the nucleus of the neighboring cell usually controls the function of the non-nuclear cell, and that is how this both of the cells they function together. And now you can also connect back to the statement, if one of the cell dies the other one also dies, because then other cell will not be able to function without the nucleus at all. And these companion cells, they are only found in angiosperm and not in gymnosperms or in pteridophyte type of plant. And the reason is also probably very clear, because the type of arrangement of sieve tube is very specific, like in case of angiosperm, whatever we are looking at. Gymnosperms, these are just walls which are perforated or porous kept, and the water moves through zigzag movement. So that is the phloem tissue in gymnosperm. And in angiosperm, we have this proper arrangement of the sieve tube along with the companion cell. Okay, any question here again? Or I will ask question, because something is coming to my mind that I should ask. Okay, so let me ask, whom should I ask? Arjun. Arjun, can you tell me? Arjun, are you there?
Yes, sir, I'm here.
Okay. So can you tell me what is the function of plasmodesmata? Quick check.
Sir, could you repeat? I couldn't hear you.
Plasmodesmata. What is the
Uh,
function of plasmodesmata?
Sir, it is the end wall of the sieve tube.
No, that is specifically called as transverse septum. It's a septum.
Sorry, sir, I don't know.
Okay, fine. Ruhan. Mr. Ruhan. I can't hear you if you are trying to speak. No. Okay, I don't know what is wrong there, but I can't hear you. Uday, what do you think? What is plasmodesmata structure? Uday? Gayab? Uday, are you there? Not there. Rishabh, what do you think?
I'm not sure, sir.
Ayyo. Anirudh,
No, yes.
okay. So it's written here. This is the plasmodesmata. What do you think is the function? The companion cells and the sieve tube elements, they maintain close cytoplasmic connection with each other through plasmodesmata. Now, what do you think would be the function? Does anyone know? I spoke about it. Adira, you want to try? Adira? Okay. Snehashish?
Yes, sir.
What do you think is the function of plasmodesmata? Are you able to hear me?
Yeah, sir.
Okay. So can you say the function of plasmodesmata in this specific case or no? Okay, I guess let me repeat that small part there. So
Yeah.
like I said, companion cell and the sieve tube structures, they are present next to each other and they have a common wall. And I also gave you analogy of the neighboring houses, and if we just remove few bricks in the common wall and we are exchanging certain materials, so these two cells, which is sieve tube and the companion cell, will have a small tubular structure present across the cell wall, which is common to them, and through that they will exchange certain materials, and that is what is known as plasmodesmata, and the function is to allow the exchange of cellular materials.
Okay.
Okay, so then I will just quickly finish this part and then we will stop after completing the phloem. So the first component that we saw was sieve tubes. The second component that we saw was companion cell. The third component of the phloem is known as phloem fibers. Something similar like xylem fibers. They are also providing mechanical support to the conducting elements, that is to the sieve tubes, uh, to hold that structure in its place. These fibers are present and they are specifically called as libriform fibers. That is just nothing but just the name to the phloem fibers, like alternative. And these fibers, they are generally not found in the primary phloem tissue. That is when the phloem has just started growing, because it does not need much of the support, because the plant height may not be so much, right? So in that case, primary phloem will not have these libriform fibers, and they are present in the secondary phloem, providing the mechanical support. So that is the phloem fiber part. Then phloem parenchyma, again these are these living cells which are thin walled. They store various materials including your resin, latex, mucilage, and so, and these are the things that you get as a secondary secretion, either through bark or through the when you pluck the leaf and so on. But yeah, these are the materials that you have present in the phloem parenchyma. The main function of the phloem parenchyma is conduction of the food in radial direction. Same like Xylem parenchyma was responsible for conduction of water and minerals in radial manner. Phloem parenchyma is also providing food in the radial direction and also the storage of food. Now, whatever extra storage, that is the in general function of parenchyma. Even in the leaf parenchyma, if there is extra food being prepared, it will always get converted into starch granules and it will get stored there itself. And the conducting elements of the phloem, like this phloem parenchyma, they are called as lepton specifically, and these includes the sieve cells and sieve tubes as well, because this parenchyma is closer to like means the conducting element of the phloem with parenchyma, that is called as lepton, along with sieve cells and sieve tube. So this together structure of the phloem parenchyma with the sieve tubes will be known as lepton. And that is just an additional information. You need not remember this, but what you should remember for sure is the function of phloem parenchyma, that is conduction of food in radial direction. Conduction of food in upright direction or vertical direction will be always by sieve tube structure of the phloem, and in radial direction it will be always by phloem parenchyma. Okay, so let me stop here. Does anyone